Hello. My name is Lars Bode. I'm part of the UC San Diego Imprint Center of Excellence in Therapeutics, and I'm specifically directing the Human Milk Analytics Score, short MAC, that I'm going to talk about now. I'll talk a little bit about why human milk, why do we study human milk, why we're so interested in that, what do therapeutics have to do with human milk, and then specifically talk about the Milk Analytics Score, what kind of services we've been developing and are offering, and how we envision this all in the future. Okay, so let's start with this paper here that we published back in 2020 in Science, a perspective paper that specifically talked about human milk and understanding the mother-milk-infant triad and why breast milk research holds important opportunity to improve both maternal and infant health. And the key figure of that perspective paper really was here shown on the right, where we're talking about not just human milk in isolation, not just mom, not just infant as a dyad, but really as the concept of human milk breastfeeding as part of a dynamic biological system. So if you look at this, you have mom, infant, human milk as the connector in between. And all of this system here is part of a co-adapting triad that is embedded in socioeconomic, cultural, behavioral, and environmental context. So you can imagine that the mother milk infant triad looks very different in the neonatal intensive care unit, either here in San Diego or anywhere else in the US, compared to, let's say, a mother and an infant, term healthy infant if you want, but now in a rural setting in sub-Saharan Africa where resources are limited. And of course, this entire triad and the context around it looks very different. So we have to take that in consideration. On the maternal side, we have fixed and modifiable factors that determine mom in terms of genetics, nutritional status, health status, and the same is true on the infant side as well. And this entire system changes over time, is highly dynamic. It changes over the course of lactation. We know that human milk composition is different from colostrum all the way out to, let's say, 6 months or even 2 years. And in fact, human milk composition changes within an individual feed. Foremilk and hind milk are very different, and there's different components that are transferred over to the infant. We also know that not only does maternal health impact milk composition, and with that, infant, but also infant health feeds back into breast milk composition and provides the infant potentially with bioactive components that are needed in a certain disease context. So leaving all this as a backbone, how do therapeutics play into this? And this is a graphic here, an image that we used for our initial application that is modeled after the science perspective paper, where we talk about the mother milk infant triad. But now, what happens if we bring in therapeutics on either the maternal side or on the infant side? So let's talk about that. Why do we think that human milk and therapeutics are connected? Why is it so important? When we think about therapeutics and breastfeeding or human milk, we often think about human milk as a vehicle, right? So is a therapeutic that we give to mom during pregnancy or then later during lactation, is that transmission transmitted from the mom to the infant? And are there potential safety concerns that if this drug, this therapeutic now makes it into the infant at whatever concentration, is that a safety concern for the baby? That's the number one thing we think about when we think about therapeutics and lactation. But we strongly believe there is other factors that we need to consider here. One is that maternal therapeutics might not necessarily make it into the milk, but they may change milk composition. And we know that milk composition is critically important for the immediate and long-term infant health and development outcomes. So is it possible that giving a therapeutic to mom, therapeutic itself doesn't make it into milk, but now it changes, for example, milk volume, or it changes milk protein composition, or it changes oligosaccharides in human milk, which then affect the baby either positively or negatively. And the third is that we have to consider human milk components that they affect pediatric therapeutics. So now we're giving a therapeutic to the baby, but there are milk components that now can change the absorption, digestion, metabolism, excretion of those specific therapeutics that we give, which then alters efficacy or safety of those potential pediatric therapeutics. So, for example, we know that certain human milk components alter or strongly actually change microbial communities in the infant gut. And those microbial communities in their function now can alter pediatric therapeutics. So, by an indirect way of human milk changing microbial communities that then affect the safety and efficacy of the pediatric therapeutics, I think there is something that needs to be studied as well. And last but certainly not least, we know that human milk components are bioactives themselves. They are, if you want, therapeutics, natural therapeutics. And our quest here is to fully understand how these human milk bioactives work on a deep mechanistic level to then use those components as templates for new therapeutics, either for the baby or potentially also for people of all ages. And just to give you a couple of examples here on how this looks like, if we take the extreme on the early life stage. So we take the preterm infant, the baby that is born too early. There is a disease called necrotizing enterocolitis, where about 5 to 10% of all very low birth weight infants that develop this often deadly intestinal disorder. We know that human milk is protective. We just didn't know what in human milk protects the preterm infant from this devastating disease. So over the years, we published a series of papers in Gut, first showing that there is a specific human milk component, an oligosaccharide called diacylgalactotetrose, or DSLNT, that indeed prevents necrotizing enterocolitis in neonatal rats. We later showed in a human cohort here in North America that indeed this specific oligosaccharide is protective against necrotizing enterocolitis in preterm infants by association. And we later confirmed this in two other studies, one in South Africa and then referenced down here in the UK, where we confirmed over and over again that this particular oligosaccharide, if it's not there in high enough concentrations, that the baby is at higher risk to develop necrotizing enterocolitis. Or the other way around, if DSLNT is there at high enough concentrations, the baby is less likely to develop this disease. So for us, that of course opens opportunities here to develop new therapeutics based on the natural template human milk, in this case an oligosaccharide. But it also allows us to develop new diagnostics to screen human milk for high or low concentrations of specific milk components at point of care. So either in the neonatal intensive care unit or in human milk banks that then can design their pooling strategies around these different measurements. At the very other end of the life spectrum, so we're now talking about people of all ages, and especially the elderly, cardiovascular disease. We lose about 18 million people from cardiovascular disease every single year, and we think that there is therapeutic potential in human milk components. So we've just published this paper here at the end of last year, where we show that one specific human milk oligosaccharide indeed reduces low-grade inflammation and then prevents atherosclerosis development in mice. And we're continuing to develop this further as a potential new therapeutic for cardiovascular disease based on human milk templates. So in other words, think about this. We're using these components, we're feeding them to babies every 2 to 3 hours. And the likelihood that there are any adverse effects if we feed this to adults is very low. And these components are now available at large scale and fairly low cost. So we're not saying that adults should drink human milk to reduce cardiovascular disease, but there are individual components that we can now use as potential new therapeutics. So this is a side to what we're really trying to do here. So the human milk analytics score, MAC, is really designed around this notion that human milk is dynamic, complex, it's unique. It's not like blood, it's not like urine, it's not a fecal sample, it's human milk. And because it's a unique tissue, it requires human milk specific collection protocols and analytical tools to accurately characterize human milk composition. And these assays all need to be validated for human milk. So you can't just send the sample out to a clinical lab that's used to work with blood or urine samples and hope that you get good results and reliable results back. So that's why we started the milk analytics score. And we do have validated assays, of course, for macronutrients, so proteins, lipids, carbohydrates, lactose, for micronutrients, minerals, trace minerals, water and lipid soluble vitamins. Oligosaccharides, of course, which came out of the interest from my own lab. Other bioactive proteins and peptides. So these are growth factors, hormones, cytokines, chemokines, proteases. Human milk has its own proteolytic activity. We do work with our microbiome center here to offer human milk microbiome analysis. Human milk is not sterile. It contains its own microbiome. And we also started to develop mouse models that are relevant for this context. So one word about this, for the mouse models, we do know quite well now how to collect milk from mice. We can either milk them, or if you need higher volumes terminal, then we can collect larger volumes from the mammary gland directly, and we analyze them. So the assays are validated for mouse milk as well. Small volumes, naturally, but doable. We also have a few genetically engineered mice. For example, we have an ST3-gal4 knockout mouse that lacks the predominant mouse milk oligosaccharide 3-sial-lactose. 3-sial-lactose is one of two oligosaccharides in mouse milk. There's only two. Humans have 150 different ones. And we also have a human FUT2 knock-in mouse that now expresses the human FUT2 gene in the mammary gland under a lactation-specific promoter. And this animal now starts to secrete the human milk oligosaccharide 2-fucosylactose, which is the dominant oligosaccharide in humans, at levels comparable to human milk. So with all these tools, the objectives are to study the effects of maternal therapeutics on milk composition, so collecting mouse milk, analyzing it for the different components, with and without therapeutic exposure, but also to study the effects of milk components in offspring microbiome. So how do certain oligosaccharides, when they're present or absent, how do they change offspring microbial communities and function? And how does that potentially change the characteristics, efficacy, and safety of pediatric therapeutics? So this is all part of what we're offering at the core. And very important, the Milk Analytics core analytical portfolio and the different models that we have been expanding over time and will continue to expand over time because MAC received also support from our university here. So it's not just mPrint supported. We did leverage the mPrint support to garner additional support from UCSD leadership. And you see that shortly after we got the mPrint CET grant, that we started what we now call the Human Milk Institute, which is really the central hub to advance human milk from molecular discoveries to local and global public health impact. And you see there's multiple different arms, different programs that are falling under this umbrella of the Human Milk Institute, and mPrint is certainly central to that. So as part of that, and with additional support from the university, we were able to hire additional staff research associates, Gaurav and Alec pictured here. We have an ICP-MS specifically for micronutrient analysis, and also an LCMS that was provided to us by Agilent to look at the water and lipid-soluble vitamins. So this core is growing and is available for everyone to utilize. While we're talking about the Human Milk Institute, every single year we have an annual symposium that is all around human milk, and we always have at least one session that is around maternal and pediatric therapeutics. We call that the mPrint session, and it's sponsored by the mPrint Hub. Thank you very much for that continuous support. But I think it's really important to highlight this, that there is not much known about human milk. It's very clear that we need to do more work around human milk, and we're trying to fill this void by not only having the first and only Human Milk Institute, so an institute at the university dedicated specifically to human milk, but also to incorporate everything we do at mPrint, not only in the Milk Analytics core and in the different projects that we have, but also spinning this forward as we're planning these annual symposium. So please save the date, March 10 to 12 next year. Usually fills up very quickly. So I leave it with that, with our vision. We envision that the Milk Analytics core becomes a comprehensive Milk Analytics core with knowledge, skills, and technology that can serve as a resource to this mPrint, but also to other mPrint CETs as they develop, as well as to the biomedical research community at large. This is not just restricted to mPrint. These services are, of course, available for everyone. And if you're interested in collaboration or analytics, please use the email address below to contact me directly. Okay, thank you very much. And I'll leave it with that.